This week covers several fundamental principles of enzymology, including their roles in reactions and requirement for various cofactors, or coenzymes. We will also address the use of enzymes as clinically relevant diagnostic tools to determine both metabolic and genetic dysfunction. We will start with some fundamental concepts of protein chemistry. First, there are 4 different levels of protein structure. Primary structure, which is simply the sequence of amino acids in a polypeptide chain. Followed by secondary structure, which refers to the organization of short stretches of amino acids into alpha helices or beta sheets. Next is tertiary structure, where you begin to see the organization of secondary structural units into domains which have specific activity. And finally, quaternary structure, which is the organization of multiple chains to generate a multi-subunit protein. As previously mentioned, the primary structure again refers to the sequence of amino acids, as illustrated here, as it's translated off the ribosome. Shown here is a common sequence of amino acids used in alpha helices, which will generate our secondary structure. Folded secondary structures are often in the form of alpha helices or in beta sheets, both of which are primarily stabilized through hydrogen bonding. The alpha helix is the most common helical structure and is found in many fibrous proteins stabilized through disulfide bonding. These structures are extensively stabilized through a hydrogen bonding network, as shown here in the dotted lines. The hydrogen bonding is between the carbonyl oxygen of one peptide to the nitrogen group on the amino acid 4 residues ahead in the polypeptide chain. Certain amino acids, such as proline, will kink the helix, interfering with its structure. Additionally, large or bulky R groups on amino acid side chains can also disrupt these spirals. Beta sheets are also highly stabilized through hydrogen bonding. Beta sheets are a more stable secondary structure, as all amino acids participate in this hydrogen bonding, as shown here in these dashed lines. The strands may be made up of 2 or more polypeptide chains, and these can be in a parallel or anti-parallel structure. This is illustrated here in the top panel. The strands are anti-parallel, meaning they go in opposite directions. Versus in the bottom panel, you have a parallel beta sheet where all strands are running in the same direction. Again, due to the increase in hydrogen bonding, beta sheets are often more stable than alpha helices. Loops and bends make up about half of the residues in a typical globular protein. These are short segments that join 2 secondary structures. Loops tend to be structurally larger than bends and are stabilized through a variety of bonds such as hydrogen bonding, salt bridges, and hydrophobic interactions. Next, the tertiary structure refers to the interactions of secondary structures to generate a motif or a domain within a protein. These fundamental regions are partially independent within the larger protein, meaning they have characteristic function. These domains are again stabilized through disulfide bonding between 2 cysteine residues, hydrophobic interactions, and ionic interactions. In this schematic here, you can see that any given protein may have multiple domains across the structure, which allow for various functions within a single enzyme. In these examples, you can see each protein having several discrete domains, either for function or targeting. For example, this transcription factor has a DNA binding domain, as well as a nuclear localization signal. In contrast, the enzyme fructose 2,6-bisphosphatase, which we'll address later in the course, has both a functionally active kinase domain as well as a phosphatase domain and several regulatory domains. The other thing to notice is that these domains can be conserved across proteins. You'll notice similarities in catalytic domains or regulatory domains. Here are 2 examples of domains in action that we'll see throughout the rest of the course, both lactate dehydrogenase and cyclic AMP-dependent kinase. Within lactate dehydrogenase, there is a specific binding site for NAD or NADP+. This binding domain is separate, shown here, from the substrate binding domain where pyruvate is bound. Similarly, we can see an ATP binding domain in this protein kinase. As all kinases transfer the gamma phosphoryl group of ATP to protein or peptides, this binding domain is conserved among many kinases. Finally, proteins that consist of multiple polypeptide chains must assemble to form a quaternary structure, as shown here with this example of hemoglobin. We'll look more closely at hemoglobin as an example of quaternary structure in the next short video, but this will be our classic example of quaternary structure proteins, where you have, in the adult form, 2 alpha globulin and 2 beta globulin chains to form the complete protein. Each of the independent subunits has its own binding site for the porphyrin ring heme. Let's finish this with a short question. These questions are similar to those that you'll see at the end of week evaluations, as well as on tests. We'll summarize each video with either a question like this or a pathway type problem, which will allow us to conceptually synthesize the material to a higher level. In this case, our question is, an alpha helical arrangement of amino acids is considered to be what level of protein structure? Primary structure of the protein, secondary structure of the protein, tertiary structure of the protein, or quaternary structure of the protein. If we remember from earlier, an alpha helical arrangement of amino acids is considered to be part of the secondary structure of the protein. This is a classic secondary structure in addition to beta sheets. And this finishes our discussion of protein structure.